Okay, welcome to this session. Together we're going to have a look at crop monitoring using dual-pol Sentinel-1 data. My name is Amalia Castro. I work giving support to the educational activities at ESA, and I will first explain to you what this exercise is about and which data and which resources you have. Then I will also give you some time to repeat the exercises on your own, and I will be available on the chat for support. So I thank as well my colleagues Magdalena and Yolanda, which will be helping me answer your questions. So don't hesitate, we are 3 people, we can take in a lot of questions, take advantage of that. Okay, what do you have? You have 2 PDFs, right? One of them is called exercise guide, and the other one is simply called exercise. What is the difference? The one called exercise is simply, how can I say, it is simply a step-by-step tutorial. It is the list of where to click, let's say like this. Whereas the exercise guide, it is different. It includes a narrative of the context for the exercise. It also has screenshots of the results, discussion, and theory reminders. So both of them are there to help you. They help you in a different way depending on how you like to work. And you will also have the recording of what I'm saying. What you see here on my screen now is the exercise guide, by the way. That is what we're going to have a look together at. Okay, so what will we be doing? You see that we will take a dense time series of Sentinel-1 data, and we will use it to map crops. So for that we need to pre-process the data. We also need to know the ground truth, meaning which crops really grow there. We also need to see the time series, and we need to apply a classification algorithm. Okay, but the most interesting part for me, and I hope for you too, is the discussion of the results. We need to be critical with our results. We need to understand what they tell us, what they don't tell us, and how to improve them. This is why you see that here I mentioned we will complement the SAR data with NDVI time series, with the rainfall data, and with information on the phenological stages of the crop. So this is essential, really, especially when interpreting SAR, which can be a bit tricky, no? So, okay, you will also learn, how, like I was saying, how to pre-process Sentinel-1 data, and you will do it in 2 ways. One is processing a single image, so a single image manually. That's not, of course, this is okay when you don't have a lot of data, and it is important to understand why each step needs to happen. But I don't think you will be processing when you do your research or your work and you have to process lots of data. Of course you do it with batch processing, right? So the first step here is simply there to explain the second one. Okay? So let's get started. Okay, so the area we are looking at is in the Czech Republic. We were choosing this area because we were limited by the choice of ground truth data. You probably have experienced as well that it is difficult to find ground truth data. So since there was the Czech Agri study, part of Sen2Agri, Sentinel-2 for Agriculture project, we already had ground truth data available there. So very handy. We just look for Sentinel-1 data as well over there, and we created this exercise. So the choice for the time of our acquisitions, of our Sentinel-1 acquisitions, which you see is rather old, no, isn't it? So December 2015 to October 2016. It is also motivated by the time when the ground truth data were acquired. It's like this. Anyways, it doesn't matter. It's just that it's not the most recent data sets, but okay, the rest is the same. So like I was saying before, you have a dense time series. You will have 24 datasets, 24 Sentinel-1 datasets, and this is a lot, of course. We will not make you pre-process all these because you would be there until the end of the event. So we have pre-processed 21 of them for you. And then you will have to pre-process yourself only 3 so you can practice. Okay? Yeah, the crops that we have are here. Winter grapeseed, winter cereals, spring cereals, sugar beet and fodder crops, okay? If you want to know more, here's the link about the project and yeah, their final report. Okay, and if you look at your folder, actually it's nice because I have prepared a lot of things for you. There is a folder called auxiliary data. In there you have the AOIs, the pins that we will use later to create the time series. You also have the training and validation data, the coordinates of the subset that you can use to pre-process those 3 data sets I mentioned before, and the color palette for the classification result, which, I mean, this is just if you want your result to have the same colors as mine. And okay, then there is also the inputs folder. It has the 3 images that I told you in the slide before that you will pre-process yourself. And then there is the outputs folder. I advise you to save your things there. This is because normally it can, with such a long training, it can be easy to lose where we are saving our data and then we don't know anymore where to download them, right? So I advise you to just put everything in your outputs folder, and I also advise you, if you want, to organize by step like this so that you can always track what result corresponds to what. Yeah. And you will also find the 21 images that I pre-processed for you can be found here as well, okay? Alright, and then there is the outputs backup folder. I think this is very useful to give because it has all the results that I created, pre-processing the exercise, and it means that if you get lost in one of the steps or if you have any trouble with the software, you can just deal with this later. At the moment, you just go get my backup result and use it as an input so that you can carry on, okay? So that's the idea. And if you want to have a reminder about SAR for agriculture, I mean SAR basics and SAR for agriculture theory, I advise you to have a look at this training. This is a training we did in 2018 together with CONAI, and the section on agriculture was taught by Professor Christian Smulius from the University of Jena in Germany, so you can use those materials to refresh your knowledge. Oh, it also has an exercise, by the way. Okay, and now in this slide, the exercise guide illustrates what you will be seeing in SNAP after you follow the step-by-step tutorial for this section, for section step 1. So I'm not gonna bore you with the details of how to open an image, because even if you are new to SNAP and to Sentinel, you find everything written in the guideline, in the document that I mentioned before, the step-by-step tutorial. So look into that, repeat it, and if you have any questions, again, use the chat. So I just want to highlight for you the naming convention. This is the standard name, and if you want to know more about the naming convention, you can refer to the user guide. The link is in the step-by-step tutorial. So this means basically Sentinel-1A, it can be Sentinel-1A or Sentinel-1B, right? So this image was acquired by A, interferometric wide swath, ground range detected in high resolution. The 1 here means that the product is level 1. S means that it is a standard product. And DV, this has to do with the polarization. So this is dual polarization, VV plus VH. And then we have the date, 2015, 11, so November, 22nd, that's the date. And then here we have the time, and so the start time and the end time. So you can just look at this to understand the date and the month and the year. And once you have opened in SNAP, when you display this little menu, you have metadata, so on, and bands. And it is in bands where you will find the image. That's where you have to click in order to display it. And what about the preprocessing? Which steps are needed, and especially why? This is part of step 1 as well, and in the screenshots that I took to explain this to you, I am using amplitude VH. Okay, so this is my original amplitude VH image, and when I open the bands folder in SNAP, which is what I was talking about in the slide before, you see that I have amplitude VH and amplitude VV. Indeed, right, as we expected from the naming convention. One SDV means dual pole for VV and VH. Okay, so the first step that we need to do is apply an orbit file. But why? Well, this is just to correct the path of the orbit from where it was expected to be to where it really was. Yeah, it is a quick one. And we do it with the whole image without subsetting it. The second step is thermal noise removal. This is done as well in a very easy way for the user because it uses a vector that is provided already from inside the image. And the next step, you see that is a subset. Why? This is because here I am processing the whole image, and this is gigantic. It is too much to go on with the rest of the processing steps if we haven't subsetted. So I just do the subset already now, and you can see that it corresponds to this tiny area of the image. You find the coordinates in the auxiliary data folder. The next step is the calibration. When we are doing the calibration, we are relating the pixel value to the radar backscatter at the level of the ground. So you see that here in the bands we do not have any more amplitude or intensity, which are basically the same. We now have sigma 0. This is again the backscatter at the level of the ground. By the way, just to tell you that the calibration tool in SNAP is able to adapt to the different sensors. So you can use it with any SAR sensor. It will adjust the settings automatically, and you do not need to worry about how to use it. You just use it and that's it. And the next step is the speckle filtering. Notice how here we have this salt and pepper effect. This salt and pepper effect comes, in fact it is random noise, and it comes from the different scatterers which are within one cell. So depending on how those different scatterers add up to each other or subtract to each other, the final pixel value will end up being abnormally bright or abnormally dark with a random pattern. Yeah, so we filter for it. And how to filter is an interesting topic because it's better to first think about what is our objective and then think about which filter serves us well. In our case, we are working with an agricultural area where we are interested in the contents of the field, and we do not want the information of the pixels at the edges of the field to influence the values within the field. Right? So we want smooth fields and neat edges. So we use the leaf filter, because the leaf filter is an intelligent filter and it is able to recognize automatically if it is in an homogeneous area, such as the middle of the field, or if it is in an edge. And if it is in an edge, it does not apply the filter. So the edge doesn't get smoothed. And after that, the next step is the geometric correction. The geometric correction is needed because notice how here we are already changing how the image looks. This is how the product is displayed in SNAP. I didn't do anything. And this is also how it is displayed already with an orientation. This matches reality, whereas this doesn't. Because radar, as you know, acquires images in a squinted way. The image is not orthogonal, so we have, for example, the urban area is here below, and this other small urban area is in the top, with the lake on top of everything. But in reality, the larger urban area is on top, the smaller urban area is south from it, and the lake, which I don't see very well because the image is a bit dark, is at the bottom. Okay, the way this works is that it uses a digital elevation model, and it uses this to project the image into the ground. But this image, did you notice how now I was searching for the lake and it's not very clear where I can see it? This image is not visually beautiful. It's not the best when we want to open the image and explore it. So we do a conversion to decibels. We use a logarithmic scale, okay, to convert the values in the image, and it ends up looking like this, much nicer. Now I can clearly see that the lake I was talking about is here and here, and and you can also see how the values in the color manipulation tab have changed, and as well the colors assigned to it when compared to how they were before here. Now we have this dB at the end. It signals it's in decibels, and it's important that you know that this step is not needed for the classification. It is needed for the visualization. However, when you are going to process your data for the classification, make sure that you apply the same process to all of them. So if either you apply this conversion to all your inputs before doing the classification, or you do not apply it to anyone, because otherwise you will have values that are different, and this is going to mess up your classification. Another thing I want to tell you before I let you start working on your own is about help and about the metadata. Help, this is a function found here, and if you are using a tool and you are curious to know how it works or how to adjust the settings, use the help, because it will open this window. You can type here on the top the tool you are interested in or the topic you are searching help for, and it will tell you, it will show you the documentation of the tool. Okay, this is very useful. I advise you to, it is underutilized in my opinion. And the next thing I want to tell you about is the metadata. We haven't really talked about it, but it's very important. And in particular, I want to highlight this section that will appear. It's called processing graph. It has node 0 and node 1. What does it mean? As you process, as you do new steps, new nodes are going to appear, and this is how the output stores its history. Okay, so when you, I don't know, come back tomorrow or in 3 months and you open a product, okay, you can see the suffix here, and it can give you an idea of what has been done to that image before, but you will not know the settings or the details. For that, you need to go into each node, and it stores everything that you put, all the settings and everything that has been applied to the image. And like we were saying, what we just did is okay for one image, and it's okay to understand things, but later on you want to apply the same workflow to a list of images, no? And how do you do that in SNAP? Well, you build a graph, which is a workflow of a series of tools to be applied in order. And here we see the same tools that we defined earlier: apply orbit file, thermal noise removal, so on. So you first need to build this graph. You will have the tabs here, and inside those tabs you can set the settings that you like, and then you save the graph as an XML. And that's the first step. And the next step is applying this graph to a list of tools. This is done with the batch processing tool. Which is this one, batch processing. It now corresponds to step 3, okay? And well, this is the tool. It has this option to add products. So you will be adding the 3 Sentinel-1 images that you have to pre-process. So they are simply the images as they are on your virtual machine. You just load them in Snap, and then after creating the graph, you load them into this tool. Then we tell the tool that the graph, meaning the workflow that we want to apply, we tell the tool which one it is, we load it in this way, and then you will see those tabs appear. It is also possible to modify your settings in those tabs if you like, okay? And the last thing you need to do is set up the outputs folder. This is critical because in the past people sometimes get lost, they lose their outputs, right? So I advise you to really set up an outputs folder inside the Snap Practical, and another folder called step 3 where you send all the outputs of these. Yeah. Alright, so after you have pre-processed the 3 outputs, you will have a total of 21 pre-processed by me and 3 pre-processed by you, so 24. They will be opened in Snap. So now we're gonna prepare to see the time series, and we need to know 2 things. One is that the time series tool works by placing a pin somewhere and extracting the time series of that pixel, and that pixel where the pin is placed. And the other one is that, of course, we could do this with a random pixel, but that has no interest for us. We would like to know where those crops are so that we can have a meaningful time series. So I did this for you because there is, based on the polygons, the ground truth polygons that we got from Checkagri, I just selected a pin inside each polygon and I saved it. I gave it a name and a color, so you can load them. This is the easiest. You can load them, and then you can use those pins to as an input for the time series tool. It is useful to know how to create your own pins, so for that you can look in the annex of this exercise guide, and everything is explained there. And how do we display the time series for each pin? Well, you can see here that I have the time series tab in Snap open, and it has this function to load your bands, which I did here. So you see them all loaded. This is the pre-processed outputs, right? The 24 outputs that you pre-processed from step 3. So after loading them, you can apply. You can click apply, and it will show this graph. This graph with the time in the x-axis, and since we have dual-pol, we have VH and VV polarization, we can select which one we would like to display by using this little filter button. So I chose VH, okay? Then it takes some time to create the time series, and then it will appear. If you have issues displaying it, can you please write it in the chat so that we are aware? And here is how it would look like. I have displayed, by using this little function, I have displayed winter rapeseed in dark pixels, so here, this one. Each point is an acquisition. And in yellow, fodder crops. So again, each point is an acquisition, they coincide in time, time is the x-axis, perfect. This is how it would look like for you. Like I was saying, if you want to know how to create your own pixels, your own pins, or how to use the time series tool to display the average backscatter over a polygon instead of over a single pixel, you can look at the annex. There's more information there. This brings us to the part that is my favorite part. It is the most interesting part, I think, so I hope you enjoy it. This has to do with the interpretation of the results. We have already got the time series and we will comment on it, and that is the reason why we start with a small theory pill, this sort of reminder from Professor Christian Smulius, who is the teacher that I mentioned a few slides earlier. And she was telling us that, in fact, interpreting the results for agriculture is very complex. There are, as you know, different backscattering mechanisms. They can be either direct or involving double bouncing. Direct, it means that the beam will come like this and be backscattered by vegetation or by the underlying ground. And the double bounce, it means that it's just going to do a step in between. It's going to bounce back from the soil, canopy, and then back, or the other way around. And the importance of those three mechanisms, the relative importance of those mechanisms depends on a couple of things. As she was telling us, it depends on the system, it means on the satellite, right? And it depends on the target, so on the soil and the vegetation. With regards to the system parameters, so the satellite, it depends on the band. Sentinel-1 is a C band, but the other bands have more penetration or less penetration. It depends on the polarization, on the incidence angle. This makes sense, right? And it also depends on, like I was saying, the target, so the soil and the vegetation in this case, because you see they have dielectric and geometrical properties. This means their structure, their orientation towards the sensor, for example, their water content is all listed here, and yeah, also for the soil, of course. Okay, so for crops and for soil. So in summary, all this interacts in a complex way, and it means that when we want to interpret our result, it is very good to know as much as possible from the situation on the ground at the time of acquisition. In terms of course, the satellite part, this is easy to know, but I'm talking about this part. We need to know rainfall, if possible. That's the reason we included rainfall, and geometrical characteristics of the crop. This is the reason for having phenology. Now, I was not in the field at the time of acquisition in 2015 or 16 for each of the acquisitions, so I just had to Google the crops, and this was to give us an idea of their structure, of how each plant grows. I mean, winter rapeseed, as you can see, totally different structure from fodder crops. So you can, of course, this is logic that it's going to backscatter in a different way. Also, the way they are planted here, in the case of sugar beet, the structure of the whole field is different than what it seems to be like here in spring cereals. It is much more dense. So it's important to know what we're looking at. Yeah, and then we were saying phenology because of course they do not look like this the whole time, right? They grow, they dry. Yeah, it is very important and very difficult to find. So what we did here was to try to find the rainfall measurements from stations around the area where we acquired the data. So in our case it was around this city. I will not try to pronounce it because I will do it wrong. And we managed to find on this website this graph of the rainfall measurements, over time. And I overlaid these red lines which signal when there was a Sentinel-1 acquisition. Why is it important? Because if, for example, it can be this one, right? So it rained, well, not too much, but okay, just before the acquisition. This means that the electrical properties of our field are going to change for the soil, right? It means that the backscatter, I can expect it to be brighter in some areas and darker at the same time in other areas, because there will be more water if it was kind of flooded, for example. Now, luckily we didn't acquire very near this acquisition. This could have caused some, it's not really noise, right? But, well, it could make more difficult the interpretation. So that's why if you see in the time series a point that is going up a lot or down a lot and you cannot explain it, it is useful to look at this sort of data to understand why there is such a weird measurement. This slide is just to show you, to compare both. For the case of sugar beet in VV polarization, it is very hard to put them both in the same scale because they come from different softwares, and this was not really a graph, it was just an image. But I tried, and the reason I tried is because it helps us understand the timing, how the timing of the acquisition matches the rainfall measurements. This line corresponds to this acquisition, the second line to the second one, and it's interesting in the case, for example, of this second one, because we can see that there was likely some rain during the time of the acquisition, and indeed the measurement is abnormally is unusually low. No, we wouldn't expect the crop to behave in that way. This is likely an outlier, and it could be caused because the interaction of the different parameters that we listed in the slide before, among which the fact that it had been raining a little bit causes this to be in the backscatter in this pixel to be lower because it was reflected away from the sensor. That could be an explanation. In other occasions it doesn't seem to have such a strong effect, like in the case of this one. Okay, this doesn't seem such an outlier. It could be again because it doesn't depend only on rainfall but on the complex interaction between all of them. This strong zigzag, when I look at this, it corresponds to this area. It was not particularly a wet period, but still we see a lot of variation, again because of the total interaction between them. Okay, this is the same for a different crop, winter rapeseed with VH, and well, you can also analyze by yourself. I just wanted to highlight this one, which is acquisition number 1, 2, 3, 4, 5, starting from the end. So 1, 2, 3, 4, 5. If my overlaying is correct, it seems to imply that the acquisition happened during rainfall moment. So that could help explain this peak. And we can as well derive it from optical data. This is what this slide is about. NDVI time series, which can be seen here in the lower part, derived from optical data, so Sentinel-2. In this occasion, though, this time series doesn't correspond to a pin over our study area because of issues with the data, the availability of Sentinel-2 data. So in the end, I just went to illustrate this concept to you. I went and asked CONAE for help and used some ground truth data from them, derived the time series using SAR in the exact same way as we have done, but from a field where soybean was cultivated. So here you can see the soybean time series for VH, polarization. And I went to look at the NDVI time series over that exact time period. And I did it in an app called EO Browser. This is also developed by ESA, by the way, so I encourage you to have a look. It's very nice for quick visualizations and some basic analysis as well. Yeah, it allows to retrieve this time series, so I did it very quickly. Now we can see a totally different pattern, right? The one from optical data is more easy to interpret. There's the growing period, and then harvesting and going down, and then again growing, harvesting, going down. The goal of this was to highlight that interpreting SAR time series is not that easy. It is not that easy to see the same pattern. Bueno, you kind of need to know what you are looking for in order to see it, because if I saw this time series by itself, I wouldn't necessarily think that this corresponds to a period of growth. It seems to do it, but it is very complex. Yeah, so it is best used in combination with other data. However, what I was saying in the slide before depends a lot on the crop. So how easy it is to interpret depends on the crop and on the pixel probably. But yeah, for example, here what I did is use winter rapeseed in our area. We are back in the Czech Republic with a VH visualization. And since I only had data for, you see I was a bit cheeky, because since I only had data for 1 year, from 15th to 16th, in order to simulate a longer time series, I just put one after the other. This is to give you an idea of how it could look like, no, for the year after. The reason I did that is because I have the phenology, and the time, the months when the crop is growing needed a different timeline than the one here. So I had to overlap two of them so as to show you in a better way how the crop grows and how it looks in the time series. It is very interesting. This one seems to be a bit more clear than the one for soybean, doesn't it? Because we do see some increase here, which is maintained. It makes us think that this is not an outlier. This is caused by the fact that there is more backscatter from vegetation that already exists since the crop starts growing. So in the beginning it's very low, and then up it starts going up, and it's kind of maintained. It is even going up more in the last stages of the plant, and this sharp decrease matches when the plant is already ripe and harvested. So we go back to the beginning, back to a lower value, and then again it seems to be increasing again slowly. So yeah, this is a nicer time series to interpret. Sugar beet also seems to be nicer to interpret, because in this occasion I didn't need to do the cheeky trick of putting one time series after the other, because the planting and harvesting fits in the timeline where we have data. So it was planted around here. We see that the backscatter is low, some ups and downs, okay, but it seems to stay low, and then it starts going up as the plant starts growing, and the backscatter from all the structure of the plant starts having more contribution, right? And then it's kind of maintained, and I don't see a drop in the harvest, but it could be this. No, because it is harvested in autumn. I do not know why exactly we do not see the drop here. I don't know exactly what was happening in the field. I don't know, maybe they harvested a bit later, or maybe there was something happening in the field that meant the backscatters kept being high. Okay, then we are back to the processing with step 5. We are going to look at multitemporal polarimetric analysis. And for that we are going to use RGBs. And for using RGBs we need to have first of all a stack created with the 24 pre-processed images. Then we can open the RGB and we can select images with VH polarization and then with VV polarization. Let's have a look. And here we have the results of my RGB. This is the window in SNAP where you can open RGBs. I did it with VH, and I used for red, green and blue, I used 9 of January, 20 of May and 17 September, okay? So I suggest you do the same, and if you do, you will see this image. It's quite beautiful, and to help us interpret it, I used Google. I went to Google, and what do we see? Well, we have, for example, this sort of gray, but kind of pinkish gray, no? Areas. Google tells me it corresponds to a forest. And then we have these rather white areas with a bit more structure. Google tells me this is an urban area. Okay. Yeah, this is as we expected because of the different backscattering mechanisms that go on in an urban area with a lot of double bouncing, so very, very bright areas, and in a forest with a lot of volume backscattering. So it will be gray but a bit more blurred, right? And then we have areas which are rather reddish. They look, well, either red like this one or pinkish, which means the red component is higher. This means that the image that we selected for 9 of January has a higher backscatter in that area, okay? So this means that on the 9th of January, for the pixel that was red, whatever was growing on that field was causing more, was backscattering more towards the satellite. What can it mean? Well, it can mean that the crop was more developed, for example, but not necessarily. It could be that it was, I don't know, the surface of the soil was rougher and was wet, so it was backscattering more back to the sensor. It could also mean that. Areas which are green, very clear, those ones, for example, again, this is areas that had higher backscatter on the 20th of May. And blue the same, blue like this one or this one, okay? In this slide, I used the same dates, but with VV polarization. And I would like to highlight 3 things to you about the interpretation. One is the forest, another one is about the urban area, and the other one, and the trickier one, is about the crops. So first, the forest. As we know, forests have a lot of volume backscattering, and VH responds better to volume backscattering. So it is not a surprise that on this occasion with VV, the forest appears less bright. Okay, that's easy. The other thing is about urban area. Some parts of it are really bright, and this would have to do with the structure of the city. Backscattering back to the sensor, I mean the orientation and the way the buildings and the roads are oriented with respect to the sensor. And the last one has to do with the crop. This is more difficult. Let's have a look at this area here, okay, which is intense red, pinkish. Let's say red for simplicity. First, within this single RGB, if it is more red and the 9th of January is the date assigned to red, it means that whatever was growing on the field on the 9th of January, it was backscattering more than later on, than in May and in September. Okay, so this is one thing. But when we compare this region with how it looked with VH, which you can do switching slides later on, you will see that it is brighter than in VH. So with VV we are measuring the vertical component of the backscatter that reaches the sensor coming from the ground. So it means that whatever was whatever was in the ground was contributing more in the vertical component than in the horizontal component for the same date, okay? For January. It would be very nice to know what was the structure of the crop at the time. It would be extremely interesting. It is also, like we said, hard to find that information, but we can imagine that this has to do with the structure of the particular crop, how it is growing. In the previous examples we have varied the date, whereas in this one I am varying the polarization channel. Okay, so in red I chose VV for a certain date, green VH for the same date, and blue I did VV for divided by VH. Okay, so VV divided by VH for the same date always. And what do we see? How do we interpret this? Well, you already know. The only thing strange, let's say, is the yellow. Why does the urban areas and the forest, but especially the urban areas, look yellow? This is because of oriented targets. Okay, so this means those oriented targets are contributing to both the vertical and the horizontal backscatter for the red and green channels, causing higher values in them. How to create this expression, by the way, if you want to divide? Well, you can do it with this little button. This step, well, it's not that interesting. You can do it if you want. It is just showing you how to compare two layers. Yeah. Now we're coming to the last part of this exercise, the random forest classification. What is a random forest? It is basically a classification and regression tree technique that takes samples of data and variables randomly and iteratively, and it generates a large group of classification and regression trees. And well, to do it, I suggest that in SNAP you close any other products that have remained open. It will clean your SNAP. It will make so that you are not confused with other datasets. But keep the stack from step 5 open. Why? Well, because we will import the training areas, which are vectors, and we will add them to that stack. Okay? And after doing that, we will save the stack again. Those training areas are needed for the random forest classifier, that's why. And then you can go to the random forest classifier tool. You can add your stack. Okay, this is from step 6.2. It's the second stack, which includes the training areas. And in the settings of the tool, we see all these. I just want to talk to you about the number of trees. Here we selected 20. Now 20 is a low number of trees. The reason we selected it is because it will produce results quickly, in under a minute. But we encourage you, maybe better after this exercise, because we are a bit short in time, you can try using other trees and then comparing the results. in general, the more trees, the more accurate are the results. But the improvement, just bear in mind that the improvement decreases as the number of trees rises. So the number of trees is typically lower than 300. By the way, in the outputs backup folder, in particular in the step 6 folder, you will find the results that I produced with higher numbers of trees. I did 50 trees, which took me 10 minutes. I did 200 trees, it took me 25 minutes of processing. And I did 500 trees, it took me 50 minutes. So do not do it now, we don't have time. Just if you want to compare the results with more trees, just open the ones I already did for you. These are the results with 20 trees. Okay, so there's the dual-pol VV and VH, and there is also VV only and VH only. Now, the classification results using single polarization channels, so those two ones, VV and VH, have been produced for you. And you can find them in Outputs Backup, Step 6, Single Polarization. So you don't need to produce them now, you can do it later if you like, but for the moment I suggest that you just open them and compare them with the dual-pol one. Okay, we can see that the one that seems to be more consistent is dual-pol. So this is a benefit of using as many data as possible. And in the next slide, I did it with 200 trees. So for example, have a look here and notice how it is speckled, right? Because, well, this is sugar beet in this case. In the next slide, it is not so speckled. See, this is what I meant. With 200 trees, the results improved. There is less speckle, and still dual-pol continues to be the best one. Well, best one in terms of how nice it looks, but I haven't validated yet, right? And here we do a sort of validation. We import the validation polygons from the CheckAgri demonstrator project, and we manipulate their transparency, looking at their labels, to see if what was recorded on the field matches the classification that our random forest classifier tells us. You can find those validation polygons in the folder OX validation data. Okay, well, this is the end of the exercise. I hope you have enjoyed it, and just reach out to us if you have any issues regarding interpretation, regarding SNAP, regarding materials, or even after the event you can always email us without problems. Thank you very much for your attention today.